I'd like to welcome everyone to our first iSTARS Health AI Summit. iSTARS is a project, a European Union funded project from the Lisbon School of Medicine, and our goal is really to capacitate our faculty with the data science and bioinformatic expertise so that we can improve our research, our biomedical and medical research in these areas. And I'll talk a bit about that, but what I'll first like to do is really to welcome all participants, you guys here, the speakers that made this program possible, and also the organizers to doing such a great job preparing all this. Our major goal, as I mentioned, is really to bring healthcare professionals, clinical and basic researchers, and also data scientists together. And we think this is very important because the healthcare professional and the researchers, they really have the data and the domain knowledge and the medical and biomedical expertise, which combined with the technical knowledge and the ability of data scientists to build and create innovative solutions based on artificial intelligence to advance medical research is really an exciting opportunity and mixture of skills and expertises that it's needed to make health AI products a reality. And we really hope you like this conference. We try to prepare a diversity of topics that would cover different areas of the medical and also biomedical research areas. And for example, we'll have expert talks from basic, translational, and also clinical researchers. So really covering the different phases and different areas where we do research and where artificial intelligence and machine learning can be applied. We will also have like some talks from industry leaders in this area. So it's really interesting and important to bring the industry perspective here and really see what kind of tools are being developed and can be used today. We'll also have a lot of iSTARS showcases. What is this? So as I mentioned, iSTARS is our team, and we're working on multiple projects in different areas, trying to tackle different problems. And we have a series of showcases where the members of our team are going to show you what we are doing and trying to motivate you to do similar things with us. And of course, we could not forget about our ethics experts and also the patient perspective. So we also bring people discussing about these topics that are so important in health research and also especially in health AI research. So without further ado, again, just wanted to welcome everyone, and we'll now proceed with our schedule. So let's get started. So I thought about opening this conference with talking a little bit about the topics that we work on. Okay, so first, about some basic concepts, and then we'll go into some examples, really cool examples of projects and studies that have been recently published so that you guys can get an idea. And then I'll leave to other people in my group to talk about the projects that we are working on and really so that they can provide more details about the type of problems that we are trying to solve. So starting from the beginning, so what is iSTARS? So iSTARS, it's a NERA-chair project funded by the European Union. And really the goal of this funding program is to develop a new area in a given faculty of university. And with that in mind, our Lisbon School of Medical Directors decided that it would be important to invest in the area of data science, more specifically in health data science, and they applied to this funding, and they got this money to start this project, the iSTARS projects, that has a series of goals, most of them related with what I mentioned, bringing together healthcare professionals and biomedical researchers, make these people talk to each other and also talk to us, data scientists, to come up with innovative solutions in the area of health data science. And of course, we also are very interested and motivated to promote the education and raising the awareness for this topic around our community, especially about the young students, but also with other researchers. And this conference is part of this of our educational plan to promote the use of data science in our faculty. So what do we do? So we collaborate with the different researchers in the generation and also the management and planning and design of different health-related data sets that the scientists need to collect in order to answer their research questions. And in terms of analytical skills, what we are really focusing on is really health data science applications, and we'll talk a bit about what that means, and also about health genomics. So these are the very two strong areas of the iSTARS team, and now we can help other researchers. So starting from the beginning, so what is data science? So data science is really the whole process of going from collecting data and acquiring data to answer a specific research question, all the way until the part where you do the data modeling and you develop the machine learning models and so on and so forth. But this really requires the data to go through a life cycle that embodies many different steps that go from data acquisition. So how do we design the data sets that we want to collect, and why do we want to collect these specific variables? How do we organize and store them in safe environments? Then how can we process the data and really make sure that we curate the data and the data is ready to be properly analyzed? After this is done, then we can go to the data exploration step. So this will be the initial step of data analysis, where we can do data visualization, we perform the initial transformations, and also do the initial more canonical statistical analysis. After that, we might want to try things a little bit more complex and more ambitious, and then go into the data modeling step and really start employing machine learning and artificial intelligence algorithms to model our data and try to answer and create models to answer our biomedical questions. So I talked before about health data science and health genomics. So, but what is that? So in our view, the way we see health data science is really the capacity to get clinical data, but also sometimes data that is generated by basic researchers in the lab together in a way that the data are integrated and they are stored in a proper place so that they can be analyzed together. So once we have this step done, then we can go into the modeling step, as I was mentioning, and really here apply machine learning models to try to answer our questions. And there are different types of machine learning models. We'll talk about that in a bit, but the most well-known are the supervised and unsupervised. with that, we can then generate the models and try to make health-related applications. The most common and perhaps the most interesting would be those related, for example, with diagnosis, where, for example, we can use our data to diagnose choose between different diseases and try to diagnose the best we can our patients. Then we also have applications around prognosis, which will be the capacity to forecast some clinical outcome, such as, for example, is a cancer going to relapse? What is the survival time expected for a given patient, et cetera, et cetera. And then finally, perhaps more ambitious, but also within reach, think about how can we develop or recommend personalized treatments to patients. So what does that mean? It means that we could be able, based on deeply characterizing the profile of the different patients, to be able to tell what is the thing that is going to work best with this particular patient, with this particular type of characteristics, being them molecular characteristics, lab results, imaging, whatever, right? So these are like the three main applications that we do. And of course, we don't want our models to be black boxes. And by black boxes, I mean things that you can just use in predictive mode and you don't get an understanding of what is underneath. But we're really looking for having knowledge production. So to really be able to look into our models, understand why a model is doing a particular diagnosis, why is making a particular prognosis. So it's really important that by the end, we can extract knowledge and really learn new things from our models and that way advance medicine. Another applications of machine learning that we have not been doing a lot, but they are equally important, are around operational capacity, okay? So we can still use machine learning not to answer medical questions or solve research problems, but instead to, for example, optimize our hospital resources, be able to predict, for example, what is gonna be the demand for the different services, how much are we gonna spend in the different services, and with that optimize and do better clinical management. So that's also one possible application of AI in health. So we talked about supervised and unsupervised learning, but what is that? So in a nutshell, supervised learning uses labeled data to learn relationships between features of the data and the labels, okay? And I brought here an example to make things easier to understand. So if we look at these old tissue slides, okay? So the goal here would be, this is the data, right? And would be, can we find the features in this tissue slide that allow us, for example, to predict whether a given tissue or biopsy is benign or it's otherwise contains cancer cells. For example, this would be an example where you have the data and you have the labels. So basically, you know that, for example, this tissue for this patient is cancerous, another tissue for another patient, it's a benign tissue. And you provide the algorithm with these examples. So you give them the example of your input data, the tissue, and also what should be the expected output or the predicted output. And what the algorithm does, it's really to learn from these images, what are the features, for example, what are the cell types that are there, how big they are, what is next to what, and with that, be able to predict whether it's cancer or benign. So traditionally, this was had to be done in an handcrafted way. That would mean that it would require a lot of expert knowledge to really tell the programmers, and you see like the person here, to know what to look for in these images. So this person would need to explicitly program what cells that are there, what are the sizes, calculate distances, and so on, all based on knowledge that would be passed to this person by some doctor or by some healthcare professional, so that the algorithm and the model knew what to look for. So this, of course, was very laborious and required a lot of domain knowledge. But on the other hand, we were able to produce machine learning models that were quite successful, accomplishing the test that they were supposed to do, okay? But unfortunately, this was not scalable, because for every new application, you need to define a new set of rules by someone that was an expert. And probably most problematic is that this kind of introduces the bias of the person that was providing the information, because you were limited by the knowledge that was already present and acquired by the community, and it would be hard to detect new signals or new patterns that could explain the test that we wanted to achieve as well. So more recently, because of that, what people start doing was to employ more complex models where we could eliminate this person in the middle. Okay, so now instead of requiring to calculate very precise and rule-based features, the algorithms become more sophisticated, and they could themselves be able to extract knowledge automatically from the images and still classify a given tissue into cancer or benign. Okay, so this it was a great advance because it allowed the automated extraction of features. So we started now to remove this person in the middle, and then we can also recapitulate things that were already known, but also discover new features that were never thought about before. The caveat here is that the interpretability is lower, because here we were defining the features by the healthcare professional, so we already kind of had a feeling of what were things that were important. And while here they are automatically found by the algorithm, and then we need to be able to interpret them and see if they make sense or not. The other problem is that this, as you can imagine, this solution requires much more data than this one. Okay, so then this is where the limitation of this, but given the amounts of data that we have and the several decades for which we have been collecting data, this might not be so much of a problem right now. So this about supervised learning. So what about unsupervised? Unsupervised, on the other hand, learns still features and patterns and structures of the data, but in data that is unlabeled. So in this case, the data doesn't need to have any specific label. So for example, here using the same example, we have a tissue slide, but now we have no label, so we are not telling the algorithm whether there's cancer there or not. And what the algorithm is gonna do, it's gonna learn the features of the images. So it's gonna try to understand by seeing many images how do images differ, what kind of features they have. And then automatically, it's gonna learn a representation of that image. And then we can ask the algorithm, what are the regions of the images that are different? For example, you can do clustering, and here, this is what it's trying to represent. So we have dots with different colors representing different patches of this image, so different squares, and it's gonna see that some squares have cancer cells, some the green dots have stromal cells, the red dots have white, so there's nothing there. So in an unsupervised way, so the algorithm can learn to recognize different aspects of the image and really distinguish differences between them. More recently, one thing that is becoming more popular, it's really the self-supervised learning. So it's still unsupervised in the sense that we don't give it labels. And the goal is still the same. So to learn representations or to understand the image, but we use tricks to learn these features of the images. So for example, we might pick a patch like this, and we are gonna remove, for example, the center. So it's gonna be empty. And the algorithm is going to learn to learn what should be the centerpiece based just on the adjacent tissue. Okay, so we trick the algorithm so that it learns how to calculate the missing piece of the puzzle, if you will. And this allows the algorithm to learn very rich features about the images and really try to understand images even when data is missing. So that's the ultimate goal, to find these encoder features, so the features that represent the image and contain all the essence of what is represented there in the image. So this is important. This is just an example. It's important to understand that this is an example based on tissue slides, but this can be anything, okay? It can be electronic health records with text. It can be lab results. It can be X-rays. It can be anything. So the algorithms are adaptable to learn the patterns in different types of data, or even combine different types of data together. So based on what we just talked about, I just wanted to bring you like a recent paper on this area to really show you what is the strength and how powerful these algorithms can be. And this is a paper from 1.5 years ago, more or less. And what they decided to do is to use this self-supervised learning technique that I just talked about to create a foundational model. And by foundational model, they mean a model that can be used for any purpose. You know, think of for example, ChatGPT is a foundational model. So it learns about all the information and can be used to answer different types of questions. And they wanted to do this, but for retinal images, okay? And they created this algorithm that I'll present in a bit, but they call it REDFound. And it's a foundational model based on retinal images that is based on unlabeled images, so just the pictures. It can learn how to interpret those images, basically. And then they also further use it on, so this model is based on 1.6 million images, so quite a lot, right? But then they use it to predict, to diagnose different diseases with labeled data, and you'll see what kind of stuff they can do. So again, what were the data in this case? So they have the different type of exams they were looking at. So this is the CFP, or color fundus photography. So they pick all 1.6 million feet of these pictures, and they created a foundational model. And they also have a second model on a different type of picture that you can take, which is the optical coherence tomography. And they also build a model based on that. And how do they do it? As I showed you before, using self-supervised learning. So if this was the original CFP picture, they kind of mask it. You see these gray areas. So they remove these areas from the input figure. And then the model needs to be able to recapitulate the original image, okay? And by doing so, if the model is capable of doing this, it means that it's really learning how to interpret this image and come up with, you know, a very accurate representation of what the image should be. After being able to do this, then they can start using the model to do cool stuff. And what did they do? So first, they did what you would expect, right? So just try to use this model to do some diagnostic model. And they were able to show with very success that they, based on these pictures from the patients, they could diagnose diabetic retinopathy, glaucoma, and also a different set of diseases related to the eye. So that was already impressive, but not really surprising. But what they did next was to push it, and then, okay, so we did the diagnosis, can we also do prognosis? And then they tried to understand or to build a model that could predict whether a fellow eye would be evolved to a wet AMD, which is age-related macular degeneration. So it's a more severe complication. And they also show that the same model could be used for prognosis, okay? So, and that's why they call it foundational. So the same model can be used for different applications. And finally, perhaps more impressive, it's really that they went a bit further and said, okay, so, but can we, other than eye diseases, can we also predict other systemic diseases just based on this data from the retina? And they showed that they were able to make successful models for ischemic stroke, myocardial infarction, heart failure, and also Parkinson's disease. So very different diseases from the heart, from the nervous system. So really impressive and really tells you how these foundational models are able to capture patterns that really allow you to have multiple health-related applications. So that's with data science. The second topic that I would like to talk today was about health genomics, which is the second big branch of our group. And it's about using genomic data to really try to unravel new biology and new medical knowledge about diseases. So the basic idea in genomics is really that we have the ability to use genomics methods to profile the patients very deeply. And we can, for that, we say that we do multi-omics. So we can do genomics, for example, to sequence DNA, transcriptomics to sequence mRNA, proteomics to quantify proteins, all on a genome-wide scale. So really have deep molecular profiles about the patients. And if you combine that with, for example, some clinical variable of interest, such as phenotypic data, whether something is going to have cancer or not. It could also be a treatment outcome, whether a given treatment is adequate for that patient or not. So then you can combine these two types of things and really go into the understanding of the underlying biology. So, and that involves finding biomarkers, finding what are the cell types that are interacting and why they are important for the phenotypes, what are the biological and cellular processes that are changing in our patients, and so on and so forth. And genomics and multiomics is particularly important because when you think about the way genes are regulated in our cells, it's a very complex process, right? So we have multiple layers of regulation. So we have DNA being transcribed into RNA, and then we have RNA being translated into proteins. And finally, proteins will go and do their job and be involved in metabolic pathways and so on and so forth to really finally perform the functions that our cells need. And really, this complexity is hard to measure and also hard to integrate. And this is where genomics can provide a big help, because the different omics methods allow you to obtain genome-wide profiles, so across all genes and across all proteins for all these different layers. So for example, you can use whole exome sequencing to detect single nucleotide polymorphisms in cancer patients, for example. Or you can use RNA sequencing to understand what are the different pathways that are being regulated at transcriptional level. Or you can use proteomics to really get a genome-wide quantification of all the proteins in your cells. So it's really what I call a genomics revolution, because it allows us to really have really detail on the molecular profiles of our patients or subjects of study to really understand better biology. Finally, one powerful thing of having all this data is to be able to integrate the different layers and really understand that one layer alone, for example, the transcriptional layer, we get some understanding of what our cells are doing. but we really need multiple layers to understand how these things are combined together to regulate cellular processes. And again, I'll just briefly mention another area that is being very exciting and in the recent years. It's about single cell genomics, which is being able to apply genomics to the single cell level. And I took here a cartoon to kind of depict what I mean by that. So if you imagine this is an organ, maybe a brain, then in the first area of next generation sequencing, we could only do bulk RNA sequencing. So we need to disassemble all our tissues, put it all together and quantify everything together. So we ended up having an average signal of what was there in our initial sample. With single cell RNA sequencing, we are now capable to sequence and characterize the different pieces alone. And then we can group them and even identify the different regions of the brain, let's say. But we do lose the spatial resolution, right? So for example, we cannot tell that the blue and the green cells are next to each other in the original tissue. However, with spatial transcriptomics, then we can combine the best of the two worlds. So we can deeply characterize our cells that compose our tissue of interest, but we don't lose the spatial resolution. And this is really like a significant advance because it allows us not only to have deep molecular profiles, but also spatial information. And to show you again, a cool example, just really recent paper from a couple of months ago, where they were precisely doing this. So they were employing this technology to study, in this case, hepatocellular carcinoma recurrence. And what they did was that they were able to show that the distribution of natural killer cells, so one type of immune cells in our body, are prevalent in specific regions of this hepatocellular carcinoma, and that can predict whether that patient is going to recur or not. And based on that, they develop a scoring system based on a machine learning algorithm that can predict HCC recurrence. And so how did they do that? So basically they have samples from the liver, right? So they have tissue biopsies of patients that recurred and patients that did not recur. And these are just the HND stainings. And then they perform spatial transcriptomics on the same samples. And that allows them to classify the different regions of the tissues into adjacent stroma, so just the region farther away from the tumor, so the invasive front or IF of the tumor and the tumor center. So here also depicted in the different colors. And then they could start asking the question, so what is changing between recurrent and non-recurrent patients in these different regions? And what they saw very clearly is that, for example, NK enrichment score was higher for these non-recurrent patients in the invasive front and the tumor center, but not in the adjacent stroma. You can see the red are the recurrent, blue non-recurrent, and you can see that only in specific regions this difference can be predicted. And they also show that for other immune cell types like T cells, B cells, and other myeloid components, they couldn't see any difference. So when they increased their cohort and they really went on to measure with immunofluorescence whether there would be like a correlation between the NK abundance and whether some patient is going to recur or not, they could clearly see that patients enriching the NK cells were less likely to recur. And they can also show that the survival is impacted by the abundance of these NK cells in a positive way. So what they can do from this? So they can, on one hand, define signatures and biomarkers that would allow them to do this. So they identified 4 and one more biomarker that, depending on their spatial expression, they could clearly distinguish non-recurrent from recurrent patients. That's the spatial biomarker identification step. Then they can go on and do a machine learning model to really devise a score based on these biomarkers. And you can see here the score, which they call the time score, varies between 0 and 1. And you can see that non-recurrent have a lower score and recurrent have a higher score. And finally, they of course need to show that their score is better than other clinical factors that are currently being used to predict whether some patient is going to recur or not. And you can see that the time score, which is the red line here, has much better sensitivity and specificity than other commonly used clinical factors or other classification systems that are commonly used like PCLC or TNM. So this really shows you how you can employ a new technology in your patient samples to really help you or help understand biology in one way, but also provide like a clinical decision support tool for future prognosis or whether a patient is going to recur or not. So I'm coming to an end here, and I really, after giving you these two examples, I hope it makes clear what kind of added value data science and genomics can bring to study human disease, and what is really iSTARS and how can we help? So we can help with data management because you can come to us and we will help you designing your data set in the sense, so what are the variables that are important to collect, what kind of format they should be, because this is all important at a very early stage so that you collect the data properly so that then it can be used for the data analysis part. And this is something that we can help you do. And also help you like how to make your data and metadata open. So please also feel free to come to us and we will work with you on making your data open when possible so that data can be reused and we can improve the overall knowledge and the reuse of data for research purposes. So we are also experts in machine learning. Okay, so as I mentioned, very focused on diagnostic prognosis and treatment recommendation models. We'll see some examples of that throughout the conference. And also on the creation of these foundational models that I mentioned today. On the genomics front, so we are also experts in the analysis of genome-wide expression data, and that includes this, but not only, so whole exome sequencing, RNA sequencing, clip sequencing to recognize RNA binding sites, RNA binding protein sites, and also proteomics. And also, of course, single-cell genomics being a state-of-the-art technology that really gives us a lot more resolution than both data. Then we are also interested in developing projects in the area of single-cell RNA-seq and spatial omics. So this in terms of scientific competencies, but of course, we also work on more transversal areas such as science communication, funding acquisition, and also recognizing the societal and ethical implications of working with health data and also in health AI projects. So we have dedicated people in our team that could help you in these areas when you collaborate with us. And of course, in terms of scientific areas, we are very flexible. We work in different areas, but our main interests and the main knowledge and background is around these three areas. So oncology, immunology, and also neurology. This would not be possible, of course, without this amazing team. So this is us, all the people that make part of iSTARS. We are all here. Please come and talk to us in the coffee breaks and lunches and dinner. We are happy to answer any questions you might have. Of course, another important part of our work is the people that we collaborate and can work with. And these are the people that we have been working with and very happy to continue working. Thanks, everyone. for the attention, and I'll be happy to take any questions you might have.
Okay. So thank you, João. Unfortunately, our next speaker, Dr. Santiago Carmona, couldn't be here. So we're going to move on with the presentation from Dr. Mariana Santos on REDCap. Mariana is iSTARS funding officer. She holds a PhD in clinical and health services research, and her expertise spans evidence-based medicine, economic evaluation, clinical trials, with a focus on critical care and chronic disease. Welcome, Mariana. Passo aqui.
So, good morning, everyone. As Beatrice already said, I'm a member of the iSTARS team. Actually, I'm responsible for the funding, the project, but I'm also help researchers that came from looking for our help on dataset planning and their dataset design. So I will talk today about the REDCap, that it's a tool that it has been implemented at the Lisbon School of Medicine. It's not available yet, but we are quite sure that until the end of this month, it will be available for the academic community. So why iSTARS got involved at the dataset planning? We think that we can help in the identification of the variables of interest for researchers, and helping also the definition of the most appropriate measures, and according to the statistical analysis plan and other more refined methods to get to their project's objectives. So the selection of the appropriate methods and the planning of actions to reduce errors and biases, it's where I can help with Dr. João, and also planning the integration of AI and machine learning techniques to according to the objectives of their projects. It's the part where we most can help when researchers came for us. So how can REDCap help me with my research? Nowadays, most of our researchers came looking for our help when they already have a data set done, and most part of them cleaned. But we are talking here about a tool that will be available for researchers to build and manage their online data collection, both as a survey mode or a database collect virtually in database in different sites at the same time. So it's designed to be a multi-centered data collection form, almost available with most of the devices researchers have in their hands at the clinical sites, or even here with us at the university. So REDCap, it's a consortium nowadays supported by the Vanderbilt University. It's present in more than 100 countries, and the REDCap is designed to be a collaborative tool. So each partner in the consortium can contribute to improve the tool. Each partner also has its own dashboard in the consortium. This is the dashboard we will have here at Lisbon, and this is the main dashboard the researchers and data collectors will see using the REDCap. So for REDCap, for data management, can reduce data entry errors as automating data collection and support a collaborative environment for data collection. It's especially useful for multi-centered projects where researchers want to do data collection in different sites simultaneously. So nowadays it's quite difficult thing to do. Is in most of our researchers opted for using the Google Forms, that it's exactly what we are trying to avoid. REDCap allows researchers to almost fully customize their forms, so adapting their data collection for their specific needs. It's a free of charge tool. The platform can be accessed online for almost any device, and researchers don't need any additional software to import or export data. It's user-friendly. It has a user-friendly interface. It can be designed by the responsible researcher, and it has it's a model like step by step. They say that because every field constructing your form in every field, you have directions to how to do it and why it should or don't have to be done. So creating a form in REDCap, it's also researchers can do it by themselves. You don't need to have any previous experience, and also let me see. Yes, you can create your form even if you have already designed your data set at a datasheet in Excel or offline. So you can just import your datasheet for the REDCap, and then the REDCap will create or almost 100% your questionnaire, your forms. So, and the same with the export, you can export your data straight for common statistical packages you may use now, and it's sufficient to, and you also are able to, researchers are also able to extract periodic reports from the questionnaire, even during data collection periods. You can also use the REDCap scheduling function. When you have more than one data collection point along your data collection period in your study, you can just schedule. It's important when you are coordinating a multi-centered study, when at other sites has specific time points to data extraction or to interview patients or whatever your project needs, and you can just previous schedule and to better organize your data collection with the other participant sites. You can also generate ad hoc reports. That's what I said before. Along your data collection period, you can extract data reports periodically from your questionnaire. You can also put some conditional logic to your questions. It's not only REDCap can do this, but it's one of the features that REDCap allows. You can also input some formulas behind any field of your form. REDCap is compliant with data protection regulation, and it is periodically revised along. I think it's even 6 months. They are reviewing data protection strategy at the REDCap consortium. And why the university is supporting REDCap in the academic environment. This is because Lisbon Medical Schools is also wants to promote the principles of minimization and confidentiality to better comply with GDPR at our projects. So when REDCap is available for the Camel and the hospital, except the hospital has already another REDCap there, we will try to promote REDCap and deny Google Forms collections and Excel collections. So we are quite sure that until the end of the month, we will already have the REDCap available, but okay, it will be an effort at the university to researchers prefer REDCap than other solutions. So the operational procedures here for FMU. REDCap will be available at FMU websites this month, I think. And first of all, the REDCap creating your forms at the REDCap will be based on 2 different stages. At the first stage, the researcher will create and test and design during as many time as the researcher needs, they can test and modify and the form in a development mode. Along this development mode periods, the researcher won't need to have its projects approved by the ethical commission, but to change it for a producer mode, yes, the researcher will have to present the approval for the ethics commission for your data collection to begin. We are trying to put some procedures that can help researchers, but also can guarantee that the university is compliant with data protection. So the thing is, we'll have a responsible research for each project. This responsible research has to be part of the university, and then this researcher will ask for permission to build your questionnaire and to create new users. And after the approval, it will be he will have to request for changing to development mode to production mode. This is what I just talked about. About project station data storage, the REDCap is not made to be a database store. It has to be like a tool to build your form, collect your data, and then you will export your data. And after, we think 1 year, it's a good period after the end of data collection, we'll try to move. I won't say delete, but I think we are going to delete your database. So you will be asking researchers to export all your data from out of the REDCap, FMU REDCap, and then we'll be asking the researcher every 30 days if they need to keep the data set or to export and delete data set from REDCap from REDCap FMU servers. The tools that REDCap will offer, it's quite an easy tool to build your form itself. So it has an online designer. You can do it from the university or from your computer at your home. That's okay for us. As soon as we have the REDCap in the Lisbon Medical School operational, we will allow you to use it from outside of the university. So to create your form, you can drag and drop your fields from your dashboard. You can also have a code book to export your data dictionary. Your data dictionary will be always based on an Excel datasheet. It's important to check all your data set and keep keeping tracks about what have been changed along your data form creation. You can also use the survey distribution function and calendar and logging collection. Also, it's not here at the university. It's not. We don't think it will be frequently used, but you can also use REDCap for randomization when you're having like a RCT, for example. Also, you can use the data distribution tool for multi-centered projects. This is when you show to the form where your data came from, and in a multi-centered data collection. For example, if you are collecting data from Hospital Santa Maria and from Hospital São João no Porto, it's important that in Porto they won't see data collected from Santa Maria and vice versa. So using REDCap, we can assure that you can control access to data by groups of users along the data collection. And then you can decide if you, at the end of your data collection, will show your full database for all your participant centers, or just keep it from the owner of the data, as I may say like this. How can REDCap help with data management and quality control? So it allows data comparison by identifying this. You as a researcher, you can monitor your data collection and compare discrepancies along data inputs. Okay, it's important to avoid, for example, duplicities or too much different values that were different from what you expect, and also allows you to comment straight in the field. You can talk to your data collectors through comments in the fields at the formulary, at the form, along data collection. You can also keep a data resolution workflow. It's when you're cleaning your data sets. You can track and you can write queries to talk to your data collector through the form. And you can specify the reasons for missing data. And so it's important. All this data management and quality control tools are much more important when you are cleaning your data set and reviewing your full data set after the data collection, but want to keep tracking about the changes you have to do at your data along data collection period. What can I recommend about REDCap? First of all, I always say to a researcher that wants to start their project to carefully think about the variables and data collection process and what you need to as a variable, and the kinds of data collection you have to really answer your project's objectives. And when you have a data collection form to truly test your tool during the development phase to avoid changes after data collection has begun. So it's important because, for example, when you have a question with multiple answers, if you have to change one of the options or the format of your question, it will be a mess for your data set if you already have been collecting data specifically in this variable, for example. Also apply validation rules and logic to your questions at the form. It's always better to validate a long data collection than to have to correct after data collection has ended. And it's important. The most important thing about the REDCap is document the user access and the data entry along your data collection period, because most of all, to be compliant with the data protection rules, you have to control access. But here we have login and security password to access data, but you have to assure every part of your research is protected. So using REDCap, you can at least be sure that to access your data form, users are fully committed with compliance and with your research. So this is the iSTARS team. Here I think I can help researchers to build and to think about their data set, but also we have our staff or other members to help to more than plan to the data collection, plan data analysis, and suggest improvements to your project. So get in touch, and thank you. I don't know if I have any question. Don't ask, don't. Okay. I don't know when it will be available, if it's your question.
Hey, hi, thank you. I'm very, very happy to hear from you. You know, I am an enthusiastic of your project and what you do. So I appreciate it, and I thank you for the help you have already given us. So I was very much excited to hear that you have data protection by design embedded in the system, in the REDCap system, yes?
Yes.
So one very practical question, and we know you have already told us that you have an audit trail and that you have the possibility to develop and test, always to comply with the GDPR that says that we have to minimize the treatment of data and only use what is necessary for the purposes of research. So my very practical question is, if you have to export the data? You have restricted access, you have the levels of access, so only those who can access the information can do so via login and password, yes? But what if you want to export the information for any purpose, to work on further projects, for publications, etc.? Is there a possibility to encrypt or anonymize the information, if it is identifiable, that has been entered into the REDCap system? So thank you. This comes out of lack of knowledge on my part. Okay, thank you.
Okay. The REDCap has, at the REDCap environment, you have different roles for each project. So you have when you are designing your project at REDCap, you will be the responsible research. That not every time the responsible research is actually the responsible for the project. Like Professor Luis Costa can have like one student as a responsible research for one project, for example. This responsible research is, or someone that the responsible research will allow inside the system, can have different permissions to do like data export, report creations, and see, you will say if your assistant as a researcher in your project can see just 1, 2, or 3 sites or the full data set, we will establish these permissions. Yes, you can separate. You as a responsible research can separate all of this. The same will be with data extraction. As a responsible research, we will allow or deny data extraction. Most of projects, data extraction keeps always an action just for the responsible research. As I'm, for example, I'm a part of in REDCap. I'm the researcher at the research staff for a project from the ICU. And this project, I'm not the responsible research because it was not me that designed the form. The responsible research for REDCap is the one that designed the form. And then you will design the form, and then you can allow someone else to be the responsible research. But first, you will be the responsible research because you are the responsible for creating the form. And when you are creating the form, you will establish permissions for different roles. Or there's the guy who will be always inputting data. There is another guy that will always see data but not input or not export. And then you will change. There are lots of different roles you can establish, but as a responsible research, you will be the one who sets responsibilities. And most of the researchers always keep this exporting function for itself.
Okay.
But as a researcher, you will, yes. But the thing is that you can the REDCap tracks everything you do. You will see who created the report, who exported in the time, the day. It will be tracking all of actions you have done at your project. As that's a guy in the REDCap at the FMU that have actually access that REDCap calls it administrator, that has the access to all your trackings in at the FMU environment for REDCap. But this normally this guy have no time to see everything that's happening because he's a IT guy. So, but at REDCap you're responsible, but not one researcher can be responsible for different projects and have different roles in different projects. You may have more than one project as a researcher, so it's fully customizable. Yes. I don't know if I have any question. It will be fully available and it's for free for students and professors and researchers. And you just have to be part of FMU and ask for the creation of a user and making a request for the university.